In the last lecture, we talked about the creation of new variation as well as the naturally existing variability. Here we will talk about the formation of base population. So we only have one outline here, which is the principles of population formation. So in the principles of population formation, there are two most widely used techniques, which is the mutagenesis and hybridization. But for the sake of this lecture, we will focus only with the hybridization process. So this process involves cross-pollination. And when we talk about cross-pollination, we cross individuals that are genetically different or with contrasting traits in order to create individuals that are heterogeneous and heterozygotes. So in the formation of population, there are four principles that you have to keep in mind, and these are the allele combination, number of parents used, the genetic combination of those parents you selected, and the time availability in doing this population formation. So allele combination, so what's in this principle? Here, the number of intermediate generations can influence the alleles obtained by the members of a population. So the intermittent generations is also influenced by the number of parents you use to form your population. So in the diagram, we have here 4 parents involved in the first generation, and using 4 parents, it requires 2 generations of hybridization in order for you to be able that all the members of the population will have the alleles from those 4 parents. So here we have P1 colored blue, and then P2 with white bordered with black, then P3 we have green, and then P4 we have light yellow. So in the first generation, of course, you cannot do 4-way cross, okay, in one go. So first you have 2-way cross between either of the parents. So here we have B1 and P2, and the other cross is between P3 and P4. So here P1 crosses with P2 produces this hybrid 1-2. So I just label it this way in order for you to track the alleles as well as the number of the parents. So here we have combination of blue border and a white inside color of the box. So that's why you have here that denotes the hybrid between these two parents. Same with the offspring of P3 and P4. So we have here the green and the yellow border. So after the first generation, of course, if you want to have the members of the population to obtain the alleles of these 4 parents, then you have to cross the offspring of those 2 crosses. So here in the second generation, we cross 2 hybrids, hybrids 1-2 and hybrid 3-4. So here in the second generation, you're confident that this individual contains at least alleles from those parents. So here we have a white color from P2, green color from P3, a border yellow from P4, and a border blue from P1. So this shows you that the number of parents used will also affect the number of generations in order for you to have the allele combinations from the 4 parents you use in to forming your population. So this is explained further this way. So in first generation you have P1 and P2, and it produces this hybrid. So we also label here a subscript that if 1 that belongs to P1 and if 2 that belongs to P2. So here you have a combination of 1 and 2 alleles coming from the 2 parents. Same is with this cross P3 and P4. You have here A3, A4, so on and so forth. And then in the second generation, you will cross these 2 hybrids, and of course this produces genotypes with unique alleles or allele combinations. For example, I have here only 2 genotypes, but there could be more genotypes produced based on the combination. So here in the genotype 1, you're confident that this individual has a mixture of alleles coming from the 4 parents. So we have here 2, 4, 1, 3. So those subscripts denotes that that allele comes from each of these parents. And same is true with genotype 2. We have 1, 3, 2, and 4. so on and so forth. So to summarize, you can have this minimum number of intermittent generation based on this number of parents. So if you have 2 parents, then you have at least 1 intermittent generation. If you have 3 and 4, the required number of generation of intermittent is 2, 5 and 8 is 3, 9 to 16 it's 4, and 17 to 32 it's 5. So bear in mind this number of parents. This influences the number of intermittent generations if you want that your population will have a mixture of alleles coming from the number of parents you used. So number of parents, that's the second principle. So here the effect of adding more parents are to increase the number of alleles in the population as well as the burdening of genetic base. But the number of parents is not a reliable measure to really have a broader genetic base of your population or to have desirable alleles, because it's the individual performance of the parents that's the most important thing here. So for example, even though you added many parents, but if you group them, some of them are contain superior traits, let's say 10% of that, and 50% have average performance, 25 have below average, and some of them the least inferior with inferior characteristics is just 15%. So it's not about the number, but it's the performance of the parents. So here you will choose either you will only intermate 10% of your individuals with superior traits, or you may include individuals with average performance. So there will be some um, uh, considerations if you want. If you focus only on improving superior traits, then you focus on this one. So there are trade-offs of using these superior traits and including some average performance lines in order for you to do your hybridization. So you have to choose whether you want their improvement or you want to increase the genetic base. So the third principle is genetic contribution of parents. So here, breeders should utilize genetically diverse parents with unique alleles to broaden the genetic base. But the caveat in doing so is that increasing the number of parents with novel alleles for the formation of base population does not always result in a population with desirable performance. This is true because we have what we call epistasis. So some of alleles, although it's novel and it's interesting, it might not be expressed because there are other genes that have epistatic effect and will not allow this novel allele to be expressed because of this epictatic relationship. So that's the only caveat. So that's why you can do combining ability test in order to check if that allele is comparable or can be expressed in different crosses of parents. So you will choose here between genetic variability and population performance. So if you want genetic variability, then as what I have discussed in the previous slide, you will either choose those 50% with average performance, or you will include some of them together with the 10% in order for you to increase the genetic variability. Or you can only choose the 10% if you want only to have this population performance. So that's the trade-off there. If you're using the 10%, then you will have less variability. But if you include some of them from 50%, then you will have genetic variability, but the performance is low compared if you only use the 10%. Therefore, as an advice, you need to have a careful selection of parental lines for population formation. Then, of course, that will guide you. Depends upon your goal. The last one is the availability of time. So the length of time to complete one cycle of selection affects the amount of genetic improvement per year. So that's true. So plant breeders may choose either of the following, either to conduct several generations of intermating followed by selection, or minimal intercrossing but more cycles of selection per unit time. So you have to choose, but most of the breeders, they choose the last option, which is minimal intercrossing but more cycle of selection, because of course, as what I have told you, that selection plays an important role, and it occupies the bulk of the breeding process. So but of course, conducting several generations of intermating is also feasible in order for you to increase the genetic variability, followed by selection. But if you are in a hurry, and of course in plant breeding, you must be you must have increase your breeding efficiency, and breeding process must be shortened. Then therefore you have minimal intercrossing and more cycle of selection, so you will have a shorter time of releasing new varieties, uh, especially if you're competing with other plant breeders or other seed companies.